Bonjour. That's the only French thing I'm going to say. I apologize in advance. I threatened to say some more, but my children said that I mustn't because it might cause a diplomatic spat. So I'm sparing you. So I was quite lucky in that I was sent the previous lectures from last week, which I did listen to, and I can follow what you say, just about. And so it allows me to actually build on it a little bit and to reinforce some of the things. And what I really wanted to talk about today was the role in which toxicology plays in enhancing our understanding of the impacts of air pollution on health, and specifically in the way in which I think it helps us not just identify problems, but think of deliverable solutions to the problems that we face. And within this context, what I'm going to focus on is toxicology, which has the human at its heart, actually studying the mechanisms which drive the adverse effects of health pollution within the human itself. Now, I thought I'd start off by saying, what do we already know? And we've kind of already iterated that. And this is just an infographic. I've taken this from the World Health Organization. It was from 2018. And we've already seen this figure mentioned, this 4.2 million deaths associated with PM2.5, predominantly long-term exposures annually. But I think at this point we should already pause and we should say, what does that number really mean? Because I find the discussions of deaths sometimes distorts our attention a little bit, because I always start from the assumption that everybody dies. Death and taxes are the only two certainties in life. So when we talk about the number of people who die, what we're really talking about in these numbers are the number of people who are dying earlier than they should have died without air pollution contributing to their premature death. So we're looking at lost years of life across the global population. And some people are going to lose quite a lot if they're particularly vulnerable, and some people may lose a year or two, and some people may lose months to weeks to days, depending on their exposures, depending on their underlying genetics. But what's more important is that you don't just die. You die of the diseases you've developed throughout your life, the biography of the diseases that you've accumulated from your midlife into your age. And so these numbers actually hide behind them, not just dying earlier, but years of suboptimal living with chronic diseases and the symptoms of chronic disease. And if there was one thing I'd like to encourage in the debate, it would be to focus on healthy living in the final third of life, rather than the absolute number of deaths. Now, the other thing I wanted to point in is that we can quite easily, and I'm sure this happens a great deal, talk about air pollution and focus on the places where air pollution is worse. And throughout this lecture course, one of the things which has been emphasized is that we've got quite a sufficient problem enough in Western Europe. We still have about 500,000 premature deaths, earlier deaths than should be happening within Europe at the air pollution levels that we see today. And I still find it remarkable that some of the most persuasive and strong statistical evidence comes from places like Canada and from Sweden, where the concentrations of PM2.5, for example, are below the old WHO guideline level and just often hovering above the new one. So these particles are having effects on the population at very low levels, and we know what they're causing. And again, I'm not going to reiterate this because we've been through it. We know that the cardiovascular causes, we know there are the sort of like neurovascular causes, we know there are the respiratory causes. I would say again, as a little caveat, yes, that these things are often based on death certificates. And death certificates are often not massively reliable in terms of cause of death. So very often when there's a nice division between these are cardiovascular deaths and these are respiratory deaths, actually, sometimes it's safer to think of them as cardiorespiratory deaths because there's quite a lot of overlap in the way in which the diagnosis is being made. For stroke, yes, definitely, but for heart and chronic lung disease, for example, there can be problems. The other thing I'd say is that these numbers exclude, completely exclude, all the new findings on the association between air pollution and neurological endpoints, such as dementia. And so if anything, we should start thinking of these numbers as being an underestimate, or indeed the new knowledge we have about the impacts of air pollution occurring very early in life, so that potentially your health, when you're 50, 60, and 70, could already be partially written in your physiology from birth and in the early years of your life, which is an arresting thought when you start thinking about how we should deal with these issues and how quickly we should focus our attention on them. And I've kind of shown this pyramid. Everybody shows this pyramid. It's one of the most famous pyramids in air pollution science. And again, it makes the point I've just tried to make, that we all focus about this, and then because we're focusing on the death statistics, we're missing all of the much larger effects we see at the population level in terms of ill health, symptoms, medication use. And then right at the bottom, there's this sort of like horrible cerise lozenge with white writing, which nobody can see. But what it talks about is the stuff which happens to us all. And I think that's another message we have to get across. Air pollution has a physiologic effect upon us when we breathe it. It causes low-grade inflammation in our lungs. And many of those effects are subclinical. The individuals who are experiencing these biological effects do not have symptoms. It's kind of almost like a smouldering ember in the fire. The accumulation, the continuation of those low-grade, if you like, adverse responses in the body over 10, 20, and 30 years play a contributing fact to driving us towards the diseases of age. Sometimes I often think about air pollution myself in terms of the concept of almost causing accelerated organ aging in the body, driving our organs towards, if you like, conditions where they're more vulnerable to disease. Now, that's my kind of background focus, and then I'm going to tell you what an ideal world for air pollution research would look like for me. And it would look like this. Undoubtedly, the field is led and has been led by epidemiologists and by the identification of associations between air pollutants and adverse health effects, both short-term effects and long-term effects. And that has created the motivation for doing something. But alongside that, we have toxicology, which is trying to actually address the how question. How does air pollution cause these effects acutely and chronically that we see within our population? In an ideal world, I would argue that the research investment and effort addressing these questions would be equally split between these two domains, so that as we saw associations, we would understand the causality of those associations, so that we wouldn't have any nasty surprises downstream. Underpinning these are measurement science and modeling, and I'm going to make a big call out here for measurement science. We often think about measurement scientists in terms of measuring pollution in the air. A lot of the problems, and I will acknowledge there have been problems with the integration of toxicology into health studies, is that we haven't applied the same rigor to the measurements which are made within those systems for understanding biological mechanisms as the measurement scientists who measure pollutants in the air have made to their measurements, and there's a great deal of scope to make that. Now, here's the problem as I see it. Epidemiology is really quite a big area of research. Toxicology is really quite small. It takes a long time. It's really, really expensive. There's toxicology in humans, and this can be integrated with epidemiological studies and panel studies by taking blood samples and urine samples and looking for biomarkers of adverse responses. But there's also toxicological models where you try to, if you like, look at air pollution samples collected in the real world and then added to systems which are trying to mimic aspects of the biological system. So taking particles and adding them to lung cells, taking particles and adding them to cardiovascular cells, for example, and trying to see if you can understand how harmful they are, how they compare with one another, because that's a huge question, and also trying to unpick the certain extent to which you can actually replicate a pathway which would actually lead to the chronic diseases we see within our populations. Now, within here, there are some really fantastic issues and challenging issues we've got to deal with. The first one is association is not causation. And there are many fantastic ways in which you can do reverse causation studies, and you can integrate genetic risk factors into epidemiological studies. But I would still advocate for the fact that if you want to know something A causes B, you should intervene in the pathway to see if that B association disappears. And that's really what toxicology needs to do. The other thing, I've mentioned it already, is that we get and we can understand the acute responses to air pollution. Where we struggle is the long-term cumulative nature of the damage that air pollution causes and how we study it within our population. To do that, we need biomarkers. Now, biomarkers both measure how much of a chemical we've taken up into our body, but also how our body has responded in an adverse way to those chemicals. So we could, for example, measure a metabolite of an organic chemical in our body as a marker of that intake. We could measure cytokines, inflammatory mediators in our blood, to see if those chemicals have caused an inflammatory effect. But most of the biomarkers we have are relatively short term. They tell us about things which have happened in hours, in days. If we go to adducts, maybe to months. If we look in DNA adducts, potentially for cancer, you can look into years of exposure. And the holy grail with biomarkers, and it was mentioned in the last talk, was really to look at whether there are long-term epigenetic gene methylation signatures of long-term exposures. Because otherwise, we cannot make that jump between what the models say you've been exposed to based on where you live or the monitors you may be carrying, and the actual amount of the chemical entity which has entered your system and is contributing to the adverse response in the body. That means we need to have something which is called an understanding of an adverse outcome pathway. This isn't very complicated at all. We just have to understand how bad stuff happens when we take in chemical toxins. And if we're going to ask that question, there's another word here which is called MIE. That's a molecular initiating event. What causes, what is the straw which breaks the camel's back? How do air pollutants cause harm? What's the first trigger for them to cause harm? And then do we actually understand the pathways that they cause harm by and how those pathways integrate with the diseases and the disease risks we see? Now that would be a challenge. But air pollution is bedeviled by this little issue here. And this is going to be a big aspect of what I'm going to talk about. It's mixtures. We talk about air pollution, unless we're talking about chemical entities like ozone or nitrogen dioxide or benzene. To me, it almost feels as though it's medieval. How much dust is there in the air? What are the number of particles in the air? These are hundreds of thousands of separate chemical entities coexisting in a particulate, vapor, and a gaseous mixture within the air that we breathe. But for simplicity, and because of the way in which we measure air pollution, we have incredibly crude metrics. Now, this makes no effect at some level. If you, for example, say that PM 2.5 is bad, then anything you do to reduce PM 2.5 should be good. But it gets more important when you begin to get down to the very lower levels we see in Western Europe, where you actually have to know which bits of PM 2.5 you need to reduce to produce the biggest health benefit. And if you don't understand the nature of that mixture or which bits within that mixture are more harmful relative to other bits, you could invest in solutions to reduce PM 2.5 mass and miss the most harmful components within it. And in fact, if in our policy areas here, we already make these compromises without the general public potentially realizing that that's occurring. Now, I can make the point with some data from London, and my data, a lot of it's going to come from London because I'm a Londoner, so just live with me. Look, this is PM10. This is a big pie of all the different chemical classes within PM10. You don't need to read this. You can see it's multicolored. There are lots of little sections to that pie. It's chemically complicated, and I'm going to tell you that that's a massive oversimplification of the chemistry of PM10 and PM 2.5, and it varies in time and it varies in space, beyond just thinking about mass. But this one I think is really much more important. This is a daily time series of PM10 measurements, and here these are chemical classes, so elemental carbon, nitrates, water, primary organic mass, across time. And I just wanted to point out that we are focused, and we have discussions about improving air quality by targeting diesel vehicles, to reduce diesel vehicles and its contribution to PM 2.5. And yet our populations don't realise that the diesel contribution to PM 2.5 is relatively small. It's this black line. It's at the roadside in London, it's about 20% of PM 2.5 mass. Yes? At the background away from roadsides in the centre of our city, it's about 5 to 10%. So a good question is, why do we target that component when it's a small proportion of the mass and not focus things which are a bigger proportion of the mass, like the secondary inorganics or the secondary organic species in the air? Now, there is a reason for this, and there's a reason that I really want us to think about moving beyond these metrics and how they potentially change the way in which we think about mitigation. Now, we've seen lots of these, and I'm not an epidemiologist, but look, this is showing you the increased risk of hospital admissions. It's from the United States for cardiovascular disease and respiratory disease. And here we have the no effect line, that 0 in the middle of both of these graphs. And over here, instead of having PM 2.5 mass, we have components within PM 2.5 mass. And it's a time series study, so it's looking at daily variations in these components versus daily hospital admissions. And so anything which is on this side of the line is having a significant contributing effect to hospital admissions for cardiovascular or respiratory disease. And if they're bold, i.e. they're not crossing this null line, they're significant associations. And we can see that some things do come out of looking at the components. And they are elemental carbon, vanadium, and nickel for this cardiovascular disease here. And over here we have elemental carbon, vanadium, and nickel again. Now this is a US study, and this is an oil combustion signature. It's the same signature we would have in Europe for diesel exhaust emissions. So already you can look at this and you can say, it's telling us that within PM 2.5, the combustion oil-derived signatures, the chemical components, seem to be more important than other components down here. And at the bottom here, we see some things which are potentially quite natural in origin, which would be very difficult to regulate. And down here, there's a meta-analysis where somebody's looked at, well, this was done by Nicole Jensen. They've looked at risk for all-cause mortality, respiratory, cardiovascular, hospital admissions, a number of studies where they've measured PM 2.5 and where they've got black carbon measurements, again, as a better proxy of diesel emissions. And where you have paired comparators, you can see the effects attributable to black carbon seem to be greater than PM 2.5. So it's telling us, within the European context, something about sources and something about exhaust emissions within Europe, and it tells us to be looking in a particular direction. Now, we've done something similar in London, and I have to tell you, whenever we do studies in London, our data don't look so nice. They always look messy, unfortunately. But I just wanted to illustrate some truisms here. Again, here we're looking at daily total mortality in London between 2011 and 2012, so it's 2 years. We have a daily time series of NOx as a marker of general traffic, carbon monoxide as a marker of petrol traffic, elemental and black carbon, diesel, copper in the air as a marker of brake wear, zinc as a marker of tyre wear, aluminium as a marker of road dust resuspension, all things which contribute to air pollution in London. And if you look across these, and what we've done is we've separated this into what we called the seasons of London, where we decided there was a cold, wet bit and there was a dry, sort of warmer bit, which is as good as we can get. And we could see that if we saw any associations, they were with elemental carbon and black carbon within, if you like, the colder parts of the year, but not with anything else. And we looked at respiratory mortality, that these are noisier, they're big wobblier because there are fewer deaths. We can see, again, it's basically elemental carbon and black carbon telling us that there's something going on with those sources. If we do the hospital admissions, this graph is horrible, but I'll take you through it quite quickly. Again, it's the same sort of thing. Here we have our metrics. Here, all we're doing is respiratory admissions, and because that varies by age, predominantly asthma in childhood, moving towards more COPD disease in later life. These have been segregated between 0 to 14 years, 15 to 64 years, and older than 65 years. And again, it's noisy, but what we see is we only see the effects in young children with the exhaust tracers. Now, that's really interesting because it's, for us, it's making a point. And these studies were published a while back, but there is a really interesting story, and nobody who's ever read these papers has picked up on it or quoted it, so I'm going to say it here so at least it's on record, yes? There were no cardiovascular signals in London in this study between 2011 and 2012. The only significant effects we found were respiratory endpoints. A similar study in London was performed between 2000 and 2005, and they showed very strong associations between particle number concentrations and cardiovascular disease. The smallest particles in cardiovascular disease, and then it's vanished. Okay? Why has it vanished? You can imagine this was very difficult for us because nobody in this area wants this signal to disappear. And we came to the conclusion there were two things which had happened. The first thing actually speaks to something which we saw in the last lecture, and that's there are fewer cardiovascular deaths. Cardiovascular disease is actually quite well managed in Europe, and the use of statins has resulted in a reduction in the number of people dying of cardiovascular disease. But the second thing is that particle number concentrations in London plummeted in 2008. Actually, they fell across many European cities, not because of any, if you like, EU or government-led policy, but because of the introduction of low-sulfur fuel, which reduced the particle distribution in the air and resulted in a significant reduction in particle numbers. It's an amazing before and after experiment that to date, nobody has actually done the science to demonstrate whether that link exists. So again, this is my plea for somebody to do that in the community. So, things are changing. We've got effects. They tell us potentially that a relatively small fraction of PM2.5 seems to be responsible for a lot of the health effects, but that's still by association. And I think it's pretty important to demonstrate to people that exhaust emissions actually do something harmful to you as an individual. I think it's important that people can take these statistics and then contextualize them into their own experience. And one way of doing that is to actually look at what happens to individuals who are exposed to the chemical mixtures that we breathe within our cities. And so I've got some data here from a series of studies I did with my collaborators in northern Sweden, Thomas Sandström, Anders Blomberg, Annelie Bendig. And in these studies, we did put, I'm going to say, happy medical students into a box on an exercise bike. And behind this wall, this is a Volvo bus engine, diesel engine. And we fed diesel exhaust into this tube, and we diluted it and then fed it into that chamber to expose them to diesel exhaust. Just once, in the first instance, for 1 hour at a relatively high concentration. You're going to fall over backwards when I say 300 micrograms per meter cubed. That's quite high, but that's the nature of doing a toxicological study. We're not replicating the world, we're replicating aspects of a response acutely to see what happens in these individuals. So, they're exposed twice, once to diesel, once to filtered air, in a randomized way. It's a bit like a drug trial, okay? It's run in a double-blinded fashion. And then to find out what goes on, we do bronchoscopies, which means that we put a fiber optic bronchoscope into the airways, and then we can flush the airways to collect cells. But we can also take biopsies from the actual physical airways themselves and then look at what's occurring in those biopsies to see if there are any adverse biological responses occurring. Now, this is an individual's airway. So here, this would be the airspace. This is the epithelium, the cells lining the airways. And under there, we have the kind of basement membrane, which the cells stick to. And in here, we have something which is known as the submucosa. And scattered throughout the submucosa, you have a variety of immune cells and structural cells. Now, in here, you'll see the little brown dots. And those little brown dots are neutrophils, which have been stained with an antibody against a protein known as neutrophil elastase, which is kind of like a flag. I am a neutrophil. And neutrophils are like one of the first waves of the acute inflammatory response to injury or infection. Now, this is the individual after air. You can see a few brown dots. After diesel, you can see there's a huge expansion in these brown dots. There's a movement of neutrophils from the blood into the tissue. So we have an active inflammation occurring in this tissue, and this is 6 hours after that 1 hour exposure. And over here, we've just got data showing, yes, this did happen statistically in all of the individuals we did this in. It was 15 in total. Now, underlying the fact that you have these changes, there is inflammation going on, and these individuals have no symptoms. They report no symptoms at all. This is all occurring, and they are unaware of it. You can demonstrate other aspects of the pathways which are occurring. So this is a cytokine associated with neutrophil recruitment into the airways and also some priming for activation. 2 cytokines, the chemokine Gro-alpha. This is staining at the epithelium after air, and then after diesel, you can see it increases. And so again, you can see in the same individuals, they have neutrophils moving into the airway, they have mediators being released into the airway, so we have an active inflammation occurring within the tissue. Now that's important, because ultimately, in all of these disease processes, there are 2 things which are sitting in the bottom of them. One of them is inflammation in its many forms, and the other one's an associated sort of pathway known as oxidative stress. Now, we talked about measuring oxidative potential in the previous talk, and I wanted just to reflect on that. Now, this, again, is going to look very complicated, but I'm going to take you through it quite slowly. Bodies, when they are stressed, do not just fall over. They adapt to the stress to the extent that they can. So let's imagine this is an airway. These are epithelial cells. This is the liquid which coats the airways. If we have something landing in our airways which causes harm, such as particulate pollution, what initially happens is that the antioxidants within this compartment provide some initial protection. If that's overwhelmed, then the cells begin to respond to the particles. But the first thing they do is not induce inflammation. They induce mechanisms to offset the damage which is occurring. They upregulate the production of antioxidants to protect them against the oxidants in the air that they're breathing. They upregulate proteins which prevent unwanted inflammation within the airways. And it's only really when those pathways have been overwhelmed, including pathways to metabolize harmful chemicals, that you begin to see a transition into a profile where you get inflammation. So inflammation is already telling you that the body has, if you like, been stressed to an extent that its normal mechanisms for protecting itself have been overwhelmed. And ultimately, if that continues, cells will die. What happens when cells die? Well, in the lung, they get replaced, yes? And with that turnover of cells, inflammation, injury, cell death, remodeling, the airway begins to change its phenotype. And part of the long-term consequences of that, it becomes poor at being a gas exchange unit. It becomes worse at actually handling infections. Now, beneath all of that, there's lots of complicated molecular stuff going on. There are particular transcription factors which are key for switching on adaptation. There's transcription factors which are very important, such as NF-kappa B, in regulating proliferation and inflammation. But when I was thinking of talking about this and I was reviewing last week's lectures, I saw that somebody had given this slide, and so I put it here because this is such a wonderful slide to show you how these pathways work. So this is a really old study by Andrew Nell's group, and he was really interested in what this was referred to as the hierarchical response, cellular hierarchical response. So he took a cell line, THP1 is a monocyte macrophagic cell line, human, and then he added increasing concentrations of diesel particles, and he kind of measured staged positions across this continuum. And what he could show was if you increased the dose of diesel particles, you got more oxidative stress. So this is a ratio. It kind of, as oxidative stress goes up, GSSG goes up, GSSG goes down, and so the ratio changes. They looked at interleukin-8 production. You can see, well, this is going down here, but already we begin to see this kicking up at 10 to 20 micrograms. He looked at heme oxygenase, which is switched on as this adaptive pathway, and you had this sort of pattern again. just beginning to come in at 10, 10's there, 10 there. So between 10 and 20, you've got this transition between adaptation and inflammation. And then over here, he looked at markers of cell death, which seem to really only become significant when you get to about 25 to 50. So he could see this sort of graded movement from initial oxidative stress through to the tissues adapting, through to then inflammation and cell death. So the reason I put it there is that we can do the same thing in tissues you've taken from the human lung. And so if we go to those individuals from that study where we took those sort of biopsies, we can actually stain them for some of these proteins. Now, NF-kappa B is associated with inflammation, and what happens is it's held in the cytoplasm, and then during an inflammatory signal, it's recruited to the nucleus to drive the transcription of pro-inflammatory genes. And so what you're seeing here is all these red dots are the movement of the NF-kappa B from the cytoplasm into the nucleus to drive that inflammatory response. And then all of these things, I always think, and that's what it looks like if you actually do the stats on it. I know these things to maybe a non-scientific audience or many these just like number plates, but these are all proteins involved in the stress response of cells, leading towards driving an acute inflammation. And so we can see that that's increased, junc's increased, AP1, which is again one of those protective transcription factors, is increased. And so we can begin to see that in humans we see the same patterns of, if you like, the pathway from oxidative stress to inflammation to injury over the long term having an effect. So this should be really easy, shouldn't it? Because now we should be able to say this is a healthy individual. If we looked at an asthmatic, all of this would be happening, only it would be much worse. Yeah? And that would be a simplistic paradigm. And that's simply not what happens at all. Because an asthmatic airway or a diseased organ is very different from a control organ. And if you don't reflect that, you won't understand the mechanisms. So again, we did similar studies, but this time we put asthmatics in the box and we exposed them to lower levels of diesel, 100 micrograms per meter cubed for 2 hours. So actually it's only about 2/3 reduced. And then we looked to see what was going to happen, and our hypothesis was everything you've just seen would just be so much worse in the mild and moderate asthmatics, and that will explain why they're vulnerable. Now, what did we find? Now, this is really complicated, but when you do a bronchoscopy, you can't keep on going into an individual and take repeat samples. You can really just go in once, yes? You get a snapshot. But when you look at biology in a snapshot, you can see the opening pages of the chapter, the mid-chapters, and almost the prologue of the book if you look at different markers. And so here we're looking at a number of things. So here in this panel, these are neutrophils in that submucosa. Here we have neutrophils which have moved out of the tissue onto the airway surface. This is ICAM1. This is one of those proteins which sticks neutrophils in the vasculature and helps them move out into the tissue. So if this goes up, this should go up earlier than these. Then we have bronchial wash myeloperoxidase. Myeloperoxidase is a protein released by neutrophils when they're activated. So getting there isn't enough. They've got to get there, then they've got to be activated. And then we have interleukin 6 and interleukin 8 because they're involved in the neutrophils getting there. So we've got a kind of a storyline here. And then we have the responses in healthy individuals after air and diesel, and then we've got mild asthmatics and moderate asthmatics and all the asthmatics together. Now, I can cut this short because we do get significant responses. We have more neutrophils in the healthy individuals who've been exposed, more bronchial wash neutrophils, we've got more myeloperoxidase, we've got more interleukin 6, but we've got absolutely no responses which are significant in any of the asthmatic subjects at all. So that's fine, because we would step back and say that's because we're looking at standard inflammation markers. We should be obviously looking at markers which are much more specific to asthma, so we can understand this. And so again, we've done the same thing here. Eosinophils, which are, if you like, the allergic equivalent of the neutrophil. Here we have them in the tissue, in the lumen. VCAM is again the equivalent of ICAM for eosinophils. Submucosal mast cells, mast cell degranulation figures, the whole thing. What do we show? Absolutely nothing. We can show that the asthmatics have elevated levels of all of these at baseline, but they're not going up after they've been challenged with diesel. So that's interesting, isn't it? Because we have to understand the mechanism, and we've kind of got a mechanism for the healthy population, but we don't have a mechanism for what's driving what's going on in the asthmatics. And that is the moment you go back to the drawing board and you think harder. Now, fortunately, not showing a change doesn't mean that things haven't changed. You're only showing no change in the things you have measured. And so a colleague of ours in Vancouver, in Canada, asked the question, well, has the airway changed in a way we haven't appreciated, which makes the airway more sensitive to the triggers of asthma? So air pollution itself is not sufficient. It is a priming agent, which then allows a secondary trigger to produce a larger effect. Now, again, this is a human exposure study of atopic individuals, so they don't have asthma, but they have allergy. And they've been exposed for 2 hours to diesel, and then after the 2 hours, an hour elapses, and then they have allergen, an allergen to which they are allergic, instilled into their airways. And again, it's a double-blinded study, so there's a saline control, and people get diesel first, or they get air first, and it switches over. Now, down here, we have a series of graphs, and they're quite difficult to follow, but they go like this. So the circles, this is filtered air, saline. Then it goes to this one, that's diesel exhaust, saline. Filtered air, allergen. Diesel exhaust, allergen. So if there was an additive effect between diesel and allergen, we'd expect there to be a pattern like that of it going up each time. So let's have a look here. This is simply what's happening with diesel, and it's absolutely nothing. They've got exactly the same response we had for neutrophils, eosinophils, IL-8, IL-5. And then they put in the allergen, and they get hints, just beginning to see hints, that the allergic airway response is augmented, but only after the allergen. But they looked at a whole host of other things. This is neutrophil elastase. Now look, we're looking for this pattern where it goes up, like a sort of gradient. And we can see, yep, that seems to be happening for neutrophil numbers. These are a special type of inflammatory cell known as CD8, 138s. They're going up. CD4 T helper cells, they're going up. In fact, that's a very big priming response. Something's going on with the helper cells. Here we have IL-4 positive cells, and so on. So the diesel isn't causing an effect that you see itself. It's showing an effect only after a secondary trigger then acts upon the airway, which has been sensitized by the diesel exposure. And I think that's really important to note. The other thing is that putting people in a box and exposing them to diesel still isn't the real world. It is for them at that particular moment, for the people doing the research, but it's not the air that we all breathe. And so this is another study, again, this was done by colleagues of mine at Imperial College, where they tried to do similar studies, but not in a box, but in the real world. And here they took asthmatic subjects, and they essentially walked them around. This is Hyde Park, and they walked around the round pond in Hyde Park, and that was low exposure, still quite high in the middle of London. And then they walked them up and down Oxford Street. Now, Oxford Street is as close as you can get to an outdoor diesel chamber in the world. At the time this study was done, you were only allowed to use diesel taxis and buses along the road. It was tall buildings on both sides, so it effectively became an outdoor chamber. And they exposed their mild and moderate asthmatics along these two conditions, and they did show real-world effects in these asthmatics. So this is looking at forced expiratory volume in 1 second, change from baseline, so these are percentage changes. These are the asthmatics walking along around the park, if you like. These are the same asthmatics walking along Oxford Street. So you've got about a sort of a 5% reduction in FEV1. What does this tell us? It tells you that the airway is just a little bit twitchy, yeah? It's irritated. Again, they were not aware of symptoms. We could measure the differences, but there were no symptoms. And the other thing they did is you couldn't do bronchoscopies in these individuals, so they looked at a sample called induced sputum, which is charming. You get people to breathe a sort of hypertonic saline, and it causes them to gag. And they cough up balls of mucus, and you can dissolve the mucus and look at the cells and the mediators within it. And they looked at one mediator here called myeloperoxidase, again, degranulated, activated neutrophils. And what you could see is they had evidence that in the upper airway, they had an active inflammation. Now, this is all interesting, but it's not what we saw in the previous study, yeah? The previous study is saying that diesel does something, and now this is telling us that the real world does something which is slightly different from diesel, which means that this paper isn't actually about diesel, it's about diesel traffic, and it's telling us that in the real world there's another mixture. Something else is related to this effect, and that becomes really interesting. Now, that's what's going on in humans. Can we understand it a bit further? One of the things that we see in the epidemiology with asthma is there's a lag, yes, for asthma. It's about 48 hours where you get the peak response. And that's telling us, a bit like the study where they put the allergen in, that something else is happening. And one of the things which people haven't really studied is how air pollution affects not just the innate, the rapid immune response, but it affects the adaptive, if you like, immune response. And one of the key cells in trying to understand how particles, when they come into the airway, interact with the adaptive immune system, which really matters in terms of asthma attacks, is this cell here known as the dendritic cell. The dendritic cell is effectively the sort of pivot point between the innate and the adaptive immune system. And so we were really interested to see whether we could understand what was going on with particles, dendritic cells, and whether they could cause changes in these T helper cells, CD4 cells, the T helper cells, that would drive them towards a particular sort of state which is likely to exacerbate asthma symptoms. So what we did is we collected myeloid dendritic cells from the blood of individuals. So again, this is human material. And we treated those dendritic cells with urban particles. Now, what happens when you do that is that they begin to mature, yes? You can see an increase in their maturation state. And then after they'd matured, we added in CD4 T helper cells from the same individuals into those mixtures and incubated them for 5 days to see what happened to those T helper cells. And what we found is that we saw increases in IL-17, interferon, and IL-33 here. IL-23P40 becomes IE-33 when you subtract one of the backgrounds. So these are cytokine patterns consistent with T helper cells becoming Th17 cells, and that's bad news if you have asthma. Yeah? But there's something else was happening, because we thought it might be interesting from previous studies that we'd done to see whether you could block that process using vitamin D. And if you look here, this is the vehicle, this is the response, and now we put in the vitamin D. From response, bang, 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 bang. So somehow, in this study, and we've got subsequent work, we can switch off the capacity of particles to actually cause this movement towards CD17 cytotoxic, if you like, damaging T cells. And again, we've actually looked at some of these mediators from these activated T cells, and we can see them again in human subjects. So these events are happening in man. The other thing we've done, which is interesting, is we've asked a simple question about symptoms. Asthmatics often complain of symptoms 48 hours. They're not going to go to hospital, but what causes the symptoms, and can we get a bit further into the diesel? So we've become a little bit interested in the way in which particles and particle constituents interact with the nerves within our airways, the nerves which could cause a bronchoconstriction, or the nerves which could cause cough and airway irritation. And what we have here, basically, is an electrophysiological experiment. We're looking at depolarization. This is a C fiber in a guinea pig, and it's been treated with PBS. And this is the same animal after a washout period. It's treated with a diesel exhaust particles, and these are depolarizations. And they're telling us, therefore, the diesel exhaust is activating that airway nerve. Now, what's really interesting about that is if we then take the diesel particle and we extract all the organic chemicals off the surface of that, so we just have a carbon core and the organic chemicals, the carbon core no longer causes that depolarization, but the organic constituents do. Again, this is really important because people sometimes talk about black carbon and elemental carbon as being dangerous. And I'm going to say I'm a little bit biased because I think it isn't dangerous at all, because carbon's quite neutral in the body, yes. It's what's stuck to those carbon particles, the associated chemicals, which are probably driving the health effects. And this tells us that it's within the organic fraction for this particular endpoint. But again, here we can then begin to dig down. How could this be happening? Well, we know this depolarization could be triggered by receptors known as TRP receptors. And if we block those receptors in both guinea pig and human nerves, we can stop this depolarization. If we come in with antioxidants, we can block this response. If we come in and we block a transcription factor known as the aryl hydrocarbon receptor, we can block this response. So we're going from saying there is a source to saying there's a part of that source which is important, and then we're saying it's causing its effects through these pathways, and we can say they are because when we block those pathways, the response that we've observed disappears. And that's, I think, about as good as you can get in terms of demonstrating causation on something which is actually related to the type of symptoms people will experience. How much longer do I have? A couple of minutes. Okay, so here's another problem we have. The other problem we have is that in the real world it's very difficult to deconvolute things. So when we talk about exhaust, it's got, VOCs, we've got small particles, elemental particles, organic particles, NO2, all in the mix, going up and down, more or less the same way. How can we tell them apart? It's very difficult to do that epidemiologically, but I just want to give you a little insight that where the maths struggles to take the responses apart, potentially the biology might be able to. So we're back to that study in Oxford Street. This time, they've taken blood samples from those individuals, and they're looking at small circulating RNA molecules within exosomes in the blood. This is a relatively new molecule type which has been identified in the body, and it's being held up as having great promise as a biomarker of adverse responses, and also telling you which organs are being affected. Now, this looks really quite pretty, and it's very difficult to read, but what it's saying is how many of the message species that we saw are associated with NO2, ultrafine particles, PM10, black carbon, and PM2.5. Overall, 53 of these molecules changed, molecule classes. But what we can actually see is there are unique small circulating RNA species which are identified with different chemical entities which co-vary in time and space in the air, which means that they have underlying different biological events occurring in the body. When you actually look at some of these are associated with pathological processes in the heart, the brain, the pancreas. So we're seeing sort of events occurring in the lung being translated through the vasculature, potentially having effects on, if you like, the external organs. So I'm going to just zip through the next bit, because I always talk for too long, and get through to some really important things at the end, which I wanted to say. And maybe at some other point I can talk about these. This is really important. If I speak to politicians now, they will tell me, I don't really care about hearing any really more bad news about this. What are the solutions? Tell me what the solutions are. And when we tell them what the solutions are, they come back and they say, tell us some other solutions which will cost less money. This is a standard pathway. And so we're caught between doom and gloom a little bit. But there are some really positive things. Where air pollution has gone down, there have been health benefits. I believe in a previous lecture you were shown the gain of life years in the population. But if you look even potentially at children, for example, these are studies from Southern California where they looked at lung growth in children in different cohorts between 94 all the way through to 2007, 2008. And this is the proportion of children with very, very small, clinically significantly small lung volumes. What you can see is if you look at these children, between 1990 to the mid-2000s, the number of children with clinically small lungs fell in California. And it fell in California because air pollution improved in California, and air pollution in California improved because they regulated to reduce emissions. They didn't persuade people to walk or cycle. They had proper regulations to force people to do something. And this takes me to my final thing, which are positive messages, which is potentially we're getting to the end of the diesel problem. Maybe we are, maybe we aren't. In London, we have something called the ULEZ. I'm sure you have similar things in cities in Paris, where we're actually restricting the most polluted diesel vehicles into the cities before they're eventually banned. This has worked, and I think that's really important to take back to policymakers. The ULEZ was introduced around 2017. These are this is, if you like, inner London predicted levels. These are actual levels. This is what happened when the ULEZ was introduced for NO2, just as a marker of exhaust. It's the biggest single change of an diesel exhaust emission signal we have seen to date, yes? And if that doesn't produce a health dividend, our politicians are going to come back to us and say, why not? Yes, so this is going to be a major issue. And of course, it has resulted in the fact that diesel is now going to be banned. I think your ban is the ban of the sale of new diesel vehicles, but not necessarily the removal of the ones which are on the road for some time. It's about 2024 in Paris, I think. It's about 2030 in the United Kingdom. And then finally, I just want to thank all the people who are involved in the work. I've covered about half of it, but the half I've covered is, I think, something I really care about, which is actually bringing a mechanistic understanding to the underpinning of the observations you see in epidemiological studies. And with that, I will say, merci beaucoup.